So this is part 2 of chapter 38.1, and we're going to start with this general overview of the angiosperm life cycle before we go into more detail here. So we again have our male gametophyte, which will be joining the female gametophyte in fertilization to form this zygote here, which is the beginning of the sporophyte generation. So this female gametophyte and the male gametophyte are the gametophyte generation. And the female gametophyte, again, is haploid, and it's located within the ovary. And the male gametophyte is located within a pollen grain, and it is also haploid. So after fertilization, we have a sporophyte in the seed. Fertilization takes our haploid, makes it diploid. So now we have a diploid little embryo plant. And this embryo plant is enclosed in a seed wall for protection. And that seed wall oftentimes will then be enclosed by a fleshy type of fruit in order to attract dispersion or animals that can disperse the seed to a distant place for growth. And then once that seed is germinated, then we have the growth of the plant into a full-grown plant. So this is the one generation, and then within this plant, within the sporophyte, we had the beginning of the next generation, the alternate generation, the gametophyte generation. So we're going to stop here and take a look at a video, or animation actually, that goes over a little more detail here.
Angiosperm alternates between a diploid sporophyte generation, represented here by the flower of the mature sporophyte plant, and a haploid gametophyte generation. Within the flower's male parts, called the anthers, are millions of diploid spores called microsporocytes. These microsporocytes divide by meiosis to produce haploid microspores. Meanwhile, a similar process occurs within the flower's female parts, which consist of one or more carpels. In this example, the single carpel consists of a stigma, style, ovary, and ovule. A single diploid megasporocyte exists in the ovule and divides by meiosis to produce 4 haploid megaspores, only one of which survives. By producing 2 different types of spores, the microspores and megaspores, angiosperms and all other seed plants are considered heterosporous. Each microspore undergoes a mitotic division and differentiation to produce a pollen grain. A pollen grain is the haploid male gametophyte called a microgametophyte. The surviving megaspore divides by mitosis to produce 7 haploid cells. One large, centrally located cell contains 2 nuclei called polar nuclei. Another cell is the egg. The 7-celled structure makes up the female gametophyte called the megagametophyte. The pollen grain pollinates the female parts of the flower by landing on the stigma. Here, the pollen grain germinates, and a pollen tube grows down the style until it meets the female gametophyte. 2 sperm from the pollen grain travel through the pollen tube and enter the female gametophyte. One fertilizes the egg, forming a diploid zygote. The other fertilizes 2 polar nuclei, forming a triploid cell. The fertilization of both the egg cell and the central cell is called double fertilization, a hallmark of the life cycle of angiosperms. The zygote, which begins the next sporophyte generation, develops into the embryo, while the triploid cell develops into the nutritive endosperm of the seed. The seed germinates, and when the sporophyte matures, the life cycle begins again.
So the credits and the location of those animations that are in this PowerPoint can be found at the end of the PowerPoint in case you want to take a look at those animations more slowly and step through them. But right now I'm going to go through a little more detailed, slowed down version of what was covered there. So in this picture here, you can see this is the male portion of the beginning of the gametophyte generation. So within the anther, we have these pollen sacs, which are also called microsporangium. And those microsporangium contain microsporocytes, which are diploid. Those microsporocytes go through meiosis and they produce 4 microspores through meiosis. And the microspores are now haploid because of meiosis. So those will develop into the male gametophyte, which is a pollen grain consisting of these 2 types of cells within a pollen capsule here. There's a generative cell, and then there's a tube cell with its own nucleus. On the other hand, on the female side, we have within the ovary an ovule. So the ovules here, there are 6 ovules within this ovary. And within the ovule is the megasporangium. So the megasporangium is the diploid or 2N form of the genetic code for the flower, for the female portion of the flower. And the megasporangium is going to go through meiosis in order to produce 4 megaspores. The 4 megaspores actually end up becoming 1 megaspore, so 3 of them die off. And so these haploid megaspores are then differentiated and through mitosis end up forming the female gametophyte. And we'll look at this in a second. So this is parallel, the male on this side, the female on this side. So what we have happen here is the pollen grains carrying the male gametophyte will attach or become caught by the stigma, and then the stigma allows this pollen grain to begin to form the pollen tube down the style. And within this pollen tube, we have the generative cell actually divide into 2 to produce 2 sperm. So these 3 nuclei essentially are going to be what travel down to the ovary. So as it travels, you can see it going and heading to one of the ovules. Okay, so now on the female side, we look at here at what happens with the surviving megaspore. So first of all, the structure of this megasporangium is this integuments, which is actually a double layer of protective covering. And that covering covers everything except the very bottom portion of the ovule. And that bottom portion is an opening called the micropyle, which is important because that is where this pollen tube is actually going to be able to enter in and cause fertilization to occur. So when we have the surviving megaspore, that megaspore actually is going to go through mitosis and differentiation. And it's actually going to go through that process 3 times so that we end up with 8 different nuclei, which will become 8 different cells. Or actually, it will become 7 different cells because one of them is going to be a double nucleated cell. So the 7 different cells we end up with from this megaspore, they're all haploid because they were produced through mitosis, but they are differentiated. So we have the 2 in the center here, which are called the polar nuclei. We have 2 that are called the synergids, and one of them is actually where the sperm will enter this whole gametophyte. The one in the center is the egg, and these ones at the top are called antipodal cells. So now this is a continuation of that process. You can see here where this is the pollen tube here coming down, and this is a close-up of what happens as it has entered the ovule with the gametophyte. So this is it releasing its sperm, and one of those sperm is going to fertilize the egg, and the other is going to join the polar nuclei and fuse together to make actually a 3N cell. So now that we have fertilization of this egg and sperm together form the zygote, which is now 2N. And here we have the other sperm joined with the polar bodies or polar nuclei to become 3N, and this is going to become what we call the endosperm. And then as this develops, we actually have the formation of our seed. And the integuments that were surrounding this before become the seed coat. The embryo grows from the zygote, and in this case these are called cotyledons, which are the beginning baby leaves of the new plant. And then surrounding our little embryo is the endosperm, which is the nutrition for this embryo. Now there's a lot of vocabulary that's new that simply needs to be memorized. And so I broke it down into this table, and I would recommend stopping this and copying it and going over it until it becomes familiar, and get a diagram of the physiology that we just talked about and walk it through until you can become familiar with these terms. Because in order to understand this reproductive life cycle, you have to understand these different terms, and they can be confusing. But one way to help separate them out is on one side, on the female side, we're dealing with a mega version of things. And on the male side, we're dealing with a micro version. So you see microsporangium, microsporocyte, microspore. Here we have megasporangium, megasporocyte, and megaspore. So, and they basically are following a very similar parallel process where we're going from the 2N or diploid cell into a 1N cell. So our 2N cell here is the microsporocyte. Over here it's the megasporocyte. And after meiosis, we end up with 4 megaspores and 4 microspores. In this case, the 4 microspores are going to continue to survive, and each one of those is going to go through mitosis and then differentiation to form the male gametophyte, which is in the pollen grain. Whereas with the megaspore, only one survives, and that one will go on to also go through mitosis and differentiation to form one 8 nucleated haploid gametophyte. On the female side, they've learned, and we're going to talk about this in the next chapter, the role of hormones in plants' behavior and function. And auxin is one of those hormones that you're going to learn about. And in this case, auxin is involved in how the megaspore differentiates into these 8 different cells. And those 8 different cells are listed right here. In the case of the differentiation with the male gametophyte, we're not going into such a complicated 8 different cells, but instead we're going just into 2 with a generative and a tube cell, and then enclosed within a spore wall, which is the pollen grain. So double fertilization is what we'll end with and start with the last portion of this lecture. We'll end this portion here just reviewing what is it meant by double fertilization, because this is something unique to angiosperms.